Buddy, so I wanted to make this recording to kind of answer some of your emails, or to answer some of your kind of common misunderstandings as pointed out in the last 2 weeks of the discussion boards. So there were some really good questions that I could tell needed some clarification, in particular relating to genetics. And since that's a really important concept, wanted to make sure we had that down. So I made a special lecture, which I will post in our Canvas soon. But that's just what I'm going to talk about today. Just again, some of the common misunderstandings that seemingly the whole class had. So this kind of includes transcription factors and activators and enhancers, as well as alternative splicing, epigenetics, how epigenetic change compares to genetic mutations, why germline versus somatic mutations make a difference, and a little bit about nucleotide excision repair. So that said, let's go ahead and get started. Okay. All right. So I'll do this. Kind of set us up. All right, so hopefully everybody can see my screen, and you might be able to see me as well. If you can't, then that's okay. It's not that important to see my face when I talk about this stuff. Okay, so let's talk about some of the points of confusion. So first off, I want to start off with transcription factors, because as we discussed in class, transcription factors or transcriptional regulation is one of the most important components of genetic regulation, basically how we get from DNA to protein. This is, in the slides, we said this was the most important part of transcriptional regulation. And really what we meant by that is it's just kind of the most widespread method of transcriptional regulation. So before we get into specifically transcription factors, I just wanted to give you kind of an overview of really what a gene looks like. So we kind of oversimplified it, but then we learned a lot more about genetic regulation. So I kind of wanted to sum that by constructing, like, based on what we have talked about in class, what we can now kind of construct a gene. So as you can see from this slide, when we look at a strand of DNA, which is shown here, we have a bunch of different components. So when we first introduced genes, we kind of introduced genes as a promoter and as the start of a gene, and then the coding region, so the nucleotides that are used to code for mRNA, and then the end of a gene being a terminator. So that is true. Every gene is a promoter and a coding region and a terminator, so a start, middle, and end. But as we elaborated on when we talked about gene regulation, the promoter is actually a lot more complex and a lot larger than we originally talked about. So again, a gene is composed of a promoter, a coding region of DNA, and a terminator. But if we were to zoom in on the promoter has a lot of components. So the promoter is made up of a core promoter, as well as basically a regulatory sequence that is upstream of that core promoter. So again, the promoter has a regulatory sequence, which usually includes an enhancer and a silencer, and we'll talk about what those do. And then right adjacent to the coding region of a gene, there is also the core promoter, which is composed of the Tata box and the transcriptional start site. So we'll talk about what each of these do. So that's the promoter. The promoter is the core promoter and this regulatory region, which has an enhancer and a silencer. The coding region is the portion of the DNA, the nucleotides that are used as a template for the mRNA. So you can remember that we use those nucleotides, the 3 to 5 prime strand of DNA, to code for via transcription. We use RNA polymerase to convert that into a pre-mRNA, which then goes through RNA processing to make mRNA, which is then translated into protein. So it's that coding region of the DNA, of the gene, that is responsible for the protein. The promoter and the regulatory sequence upstream, that all kind of controls whether this process, transcription and translation, will actually take place. And the terminator is just like, that's the end. All right. So looking more specifically at that gene, which again, this is looking at the entire gene, there's a couple components. So like we said, we have the promoter, which collectively I have in green, which has our core promoter, which is made of the TATA box and the transcriptional start site, as well as upstream of that, we have the enhancer and the silencer. So in green, we have our promoter. Our coding region is shown in blue, and the end of the gene is shown in red. So we know that RNA polymerase, we've talked about that. That is the gene that converts DNA into RNA. RNA polymerase binds to our transcriptional start site. So the function of the TATA box, which again is called the TATA box because it's just composed of a bunch of T's and A's. Transcription factors, kind of general transcription factors, bind to the TATA box. So what's the point of these transcription factors? They basically kind of like hold on to RNA polymerase, and as we will see, when necessary, they will kind of like let RNA polymerase do their job. So RNA polymerase binds to the transcriptional start site. Transcription factors, general transcription factors, bind to the TATA box. And then we have the enhancer and the silencer. So first let's talk about how we turn a gene on, right? So let's say that we do want to make a protein out of this gene. So if that happens, what we have are little proteins called activators. And these activators bind to, within the regulatory sequence of a promoter, they bind to what is called an enhancer. So the enhancer is a region of DNA. It's a region of nucleotides. And an activator, just like transcription factors, an activator is actually composed of a protein. It's a DNA binding protein. So it binds to the enhancer, which is a region of DNA. What happens when that activator binds to the enhancer is it recruits another protein, and this is called DNA bending protein. And you can kind of see what happens when the activator binds to the enhancer. The DNA bending protein literally causes DNA to bend over, and that allows the activator to associate with the transcription factors. So again, we have our RNA polymerase bind to the transcriptional start site. Our transcription factors bind to the TATA box. And when we have an activator bind to the enhancer, that recruits that DNA bending protein, and then the activator is able to associate with the transcription factors. And if we get the activator associating with those transcription factors, what that does is it allows RNA polymerase to release from this complex and transcribe that coding region into pre-mRNA, which will then be processed into mRNA, which then can be translated into protein. So again, unless we have that activator associate with those transcription factors, we won't get transcription. So again, that's kind of the importance of the TATA box, that we get those transcription factors, and the importance of the enhancer region is to basically have that activator turn on RNA polymerase. Okay, so what about that other region of DNA, the silencer? Well, basically that's just the opposite of what an activator and enhancer complex does. If you have a repressor, same thing will happen. RNA polymerase is always going to be recruited to the transcriptional start site. You're still going to get general transcription factors recruited to the TATA box. But what would happen is if the kind of microenvironment says that we don't want this gene turned on, what would happen is instead of recruiting an activator for this gene, we would recruit what's called a repressor for this gene. And a repressor functions very similar to an activator. Instead of binding to the enhancer, it binds to a silencer, which is just another region in the DNA, recruits that DNA binding protein. But when the repressor associates with the transcription factors, what happens is RNA polymerase is not released and it is stopped. And what this means is that we will not get transcription of that gene. So basically that gene is turned off. So essentially the way you can think about this is that activators will turn on transcription and repressors will turn off transcription. All right. So this is just a summary slide of what we now know with general transcription factors, how genes are turned on or off at the transcriptional level. Remember that a gene is composed of a promoter, the coding region, and the terminator. It's just that we know that the promoter has a lot more elements than we previously talked about. The promoter is composed of the regulatory sequence, which includes basically the on switch, which is the enhancer, or the off switch, which is the silencer. And then closer to the coding region, you have the core promoter, which includes the TATA box, which is where the general transcription factors bind. And then the transcriptional start site, which is where you have your RNA polymerase bind. So that just gives us a better idea of how the kind of at the gene level, the DNA level, how we can regulate whether a gene is turned on or off. All right. Now to talk a little bit about alternative splicing, which also seemed to be a pretty big point of confusion. This is a picture taken directly from a lecture. So I just kind of want to reiterate what was said in lecture. And I'll kind of go on to explain it a couple different ways. So if you remember, hopefully, because we just talked about it, that you have DNA, and that in that DNA, you have both coding and non-coding regions. So those coding regions are called exons, and the kind of intergenic non-coding regions are called introns. So you can see that in this gene, you have 5 exons. So that means 5 regions of DNA that are going to directly contribute to your mRNA. Okay, so as you know, what happens is this gene is transcribed into pre-RNA. And remember, pre-RNA is RNA that is not processed. So what has to happen with RNA processing, remember we add a cap and a tail, but the other thing that happens during RNA processing is that RNA is spliced. So when we first introduced the concept of splicing, what we said is that basically just the non-coding regions or those introns are removed. So that does happen. So you can see that when in this picture, when we transcribe DNA into pre-mRNA, you can see that the intronic or non-coding regions are shown in pink. So when splicing occurs, those introns are removed. We do splice those out. But another thing that happens for several genes is that when those introns are spliced out, depending on what gene product we want, we can basically include or exclude or basically splice out different exons as like with the introns. So for example, if our protein that we want is coded for by the combination of exons 1, 2, 3, and 5, what can happen during splicing is you can splice out all the introns, but also splice out the 4th exon. And then you would get a combination, which is shown on the left, which is an mRNA with the exons 1, 2, 3, and 5. So let's say that combination of exons codes for protein 1. All right, so the exons 1, 2, 3, and 5 code for protein 1. Now let's say something changes and that gene needs to produce a different gene product. So what can happen is you can take that DNA, and again, when you go through splicing, when you splice out the introns, let's say protein 2 is coded for by the exons 1, 2, 4, and 5. And so what happens is you can just splice out the third exon with that, and you would have that combination 1, 2, 4, 5. Because that's a different combination of exons, which remember, exons are composed of nucleotides. So if you have a different combination of nucleotides, that is going to lead to a different combination of amino acids, which is a different protein. So you can kind of see this. I want to say thank you so much to Karina, who posted this in the discussion board, which is just like an excellent explanation of alternative splicing. So she said that if we first get this pre-mRNA transcript, right? So this is pre-splicing. And let's say the pre-mRNA has this sequence, which is like green, blue, red, yellow, and pink. Those are the exons. And then the intronic regions are those little purple dots. What can happen is, let's say one protein includes this like green, blue, yellow, and pink. And so all you would need to do to get that is basically when you remove the purple dots, you just remove the red dot as well. So that sequence of amino acids is green, blue, yellow, and pink. So that's a sequence of amino acids that specifies protein 1. Alternatively, what you could do, so this is called alternative splicing, means that it's giving you multiple options. If you have multiple exons, you have multiple options for amino acid sequences. So what you could do here is, instead of excluding the red exon, you could exclude the yellow exon. And this would leave you with green, blue, red, and pink. So again, this is a different sequence of amino acids. So that gives you a completely different protein. So it just means that if you have a combination, so I guess what alternative splicing means is if you just have like various amounts. So in this case, Karina gave us a DNA sequence with 5 different exons. That gives you 5 exons that you can use in different combinations. And those different combinations will give you different amino acid outcomes. And different amino acid outcomes means you're getting different proteins. So this is how one gene with multiple exons can code for different proteins. So you have multiple exons, you can have multiple combinations of those exons, what are called splice variants, and that would give you different proteins. I also included a couple videos here. I'm not gonna force you to watch them through this presentation, but again, these are linked in the PowerPoint I posted on Canvas, this PowerPoint, and they do a really good job explaining it as well. I also copy and pasted one of my responses to the discussion board, which is just asking you to remember that kind of like in general, with transcription and translation, what you're doing is you're going from DNA to RNA to protein. And if you remember, in DNA, you have exons, and those exons are moved into mRNA. And right, the mRNA codes for codons. So if you differ in exons, you're going to differ in codons. And if you differ in codons, you're going to differ in amino acid sequence. And so basically what that means is if you jumble up your exon sequence or included exons, you're going to jumble up your amino acid sequence. So that gives you, again, a lot of possibilities for protein outcomes. So again, I do recommend you watch these videos as well. They can help. But the overall kind of like what I want you to get from alternative splicing is that it allows you to have one gene code for multiple proteins. And that's kind of the overall takeaway from alternative splicing. All right. Now, let's talk about epigenetics. And I like the idea of revisiting this. Someone asked a really good question that was, well, several of you had the question of like questions related to not really understanding how you can have a change in phenotype without changing the genotype. And another kind of to elaborate on that, somebody asked kind of the difference in effect when you compare genetic mutation to epigenetic change. So really, really good questions. So I wanted to specifically address those. So in order to do that, I kind of want to compare them. So just a reminder that with normal transcription and translation, what happens on a general level is we have a strand of DNA, which is double-stranded, 5 to 3 and 3 to 5. And we use the 3 to 5 prime strand as our coding strand. And what that means is RNA polymerase reads the 3 to 5 prime strand into a 5 to 3 prime strand of mRNA. So that's read by complementary base pairing, meaning that if you have a T in the DNA strand, that'll lead you to its complement, which is A in the mRNA. If you have an A, its complement in RNA is U, C, G, C, G, C, G, et cetera, et cetera. So you use complementary base pairing to go from DNA to RNA. And then you have, via the process of transcription, that means you have a strand of mRNA. And then we can use the codon table shown below to basically figure out what these codons, what amino acids they code for. So hopefully by now you have memorized that AUG, the first codon shown here, codes for the amino acid methionine. And then we can look up our second codon, GGG, codes for our second amino acid, which is glycine. And then CGU codes for arginine. And then we get to this codon, UAG, which turns out codes for a stop codon. So we would actually have 3 amino acids in this polypeptide. So that's normal transcription and translation, going from DNA to mRNA to protein. And from DNA to RNA, it's via complementary base pairing. And then based on those complementary base pairs, you get mRNA, which the sequence of your mRNA directs which amino acids are going to be in a sequence and thus what protein you're going to have. So that's normal transcription and translation. When we have a genetic change, which we talked about last week, I'm just going to use the example of a missense genetic mutation, which means, remember, missense means mistake. So in this case, you can see what happened is we would have a genetic mutation that changed this C into a T. So that is a change in the genotype. This is very important. So a genetic mutation is a direct change in the nucleotide sequence, and thus that is a change in the genotype, which genotype is nucleotide sequence. So a genetic mutation, this is again a base substitution. We had that C substituted for a T. That changed our nucleotide sequence, changed our genotype. What that means is when we used complementary base pairing to create mRNA, our mRNA sequence was different. And you can see AUG was not changed. We still have our methionine. But you can see that second codon was changed from GGG, which coded for glycine, to GAG, which codes for glutamate. So this did change our amino acid sequence. So with genetic mutation, what's happening is you're changing genotype, and this does change your phenotype sometimes. So the reason I use missense mutation, just a reminder, if you're qualifying a phenotype as being the amino acid sequence, then yes, this would mean that your phenotype was changed. But remember that true phenotypic change depends on a lot of things. So if glycine and glutamate were both like acidic, then your resulting protein might actually not have changed. If like glycine was acidic and glutamate was basic, then probably the resulting protein would change and you would see a change in phenotype. So the change in phenotype, whether there is a change in phenotype, depends a lot on what amino acids are changed and how this affects the protein structure. But regardless, you are definitely seeing a change in genotype. All right. If this occurs in the germline, it can be inherited because you're changing the DNA. That DNA can be inherited if it's in the germline, which we'll talk about next. Okay, so that's genetic change. Now let's look at epigenetic change. So again, on the left, we're looking at normal transcription and translation. What happens with epigenetic change is you have something called, they're called like methyltransferases. It's like a really complicated name. We don't even know about it. But you have these molecules that basically add methyl groups. In eukaryotes, this is primarily or the nucleotides is primarily methylated are cytosines, which is why I have it there. And you can see what happens is that with epigenetic changes, notice that our nucleotide sequence is not changed. Here, with normal transcription and translation, you have TAC, CCC, GCA, ATC, CTG. Here, you have the exact same sequence, TAC, CCC, GCA, ATC, CTG. The sequence of nucleotides has not changed. That means that with epigenetic changes, your genotype is not changed. Your genotype is unchanged. However, what is changed is just a little bit of the chemical structure of those cytosines. And the implications of this are that when RNA polymerase comes over to the sequence and tries to transcribe it into mRNA, it doesn't recognize those cytosines. So even though you still have cytosine, it's not being recognized by RNA polymerase. So RNA polymerase can't read this into a complementary mRNA sequence, and therefore that gene is not transcribed. So this is giving you a change in phenotype, right? In this case, you're getting a polypeptide, you're getting a protein. But if you have a lot of methylation, your genotype is not changed. Your genetic sequence is still the same, but your phenotype has changed. Like the gene's turned off, so your phenotype is changed. So this is how you can get a phenotypic change without changing the genotype. There is chemical modification, but it's not changing the nucleotides. This can, just like genetic mutation, be inherited if it occurs in the germline, which again we'll talk about. This is just a slide showing all three of these: normal transcription and translation, genetic mutation, which again is going to be a change in genotype and possibly a change in phenotype depending on the mutation. And then epigenetic change is not changing the genotype. Again, we didn't change the sequence. We chemically modified it, but it didn't change it. And that did change the phenotype. Both of these can be inherited if they occur in the germline. Not necessarily going to affect anything if it occurs in somatic cells, as we'll talk about. Or at least it's not heritable if it occurs in somatic cells. All right. I did have a question that asked about how this can be reversed. So just like you are referring to epigenetic changes, not necessarily genetic mutations. Epigenetic changes, just as they can be added by those methyltransferases, you have other molecules called, I think, like TACs, if I remember what I was just told. And these come and just can remove methyl groups. So just like we can add methyl groups, you have enzymes that can remove those methyl groups. So that's why methylation can basically be changed anytime, added or removed. And I also wanted to add that there's a little confusion about this as well. Epigenetics refers to basically having DNA transcriptionally active or inactive without changing the nucleotide sequence. So again, epigenetics, typically what we talked about before this, we talked about methylating individual nucleotides, so cytosines. And again, if you methylate those cytosines, RNA polymerase is not going to be able to recognize the sequence, and therefore that gene is turned off. All right, so again, methylation will reduce transcription in that case. However, like we talked about in class, you can also have methylation occur not only of individual nucleotides, but you can have methylation of histones, which are the proteins that DNA is wrapped around. And what happens if you methylate histones is DNA is wrapped really, really tightly around those histones. And if DNA is wrapped tightly around histones, then it is inaccessible to DNA polymerase. So again, this would basically reduce the ability of RNA polymerase to access that DNA, and therefore transcription would be turned off. So regardless, epigenetic change, methylation, is going to reduce transcriptional activity. I do want you to remember that this can, it basically can affect histones, methylation of histones, which basically silences DNA. Or you can have methylation of individual nucleotides, which would, again, turn a gene off. All right. So that's epigenetics. Now, let's talk about the difference in germline and somatic mutations. So there was a lot of confusion here, but I think it's because we kind of needed to call pretty far back based on what we were talking about when we talked about inheritance. And honestly, I get it. When we talked about inheritance, that was before everything went crazy. So I do want to just kind of tie this all in together, right? Because we're talking about genetic mutations. And we mentioned kind of the impact of these genetic mutations, inheritable versus non-heritable, and what can affect that. And I just want to remind you that in the body, there are two types of cells, and those can be classified as somatic cells or sex cells, which are also called gametes. Somatic cells are all the cells in the body that are your non-sex cells. So these would be your diploid or diploid, however you want to say it, cells. So like your skin cells, your muscle cells, your nervous cells, basically all of your cells except for your sperm or your egg. Your sex cells, which are again called gametes, are created by what are called your germline cells. So these are stem cells in your body that produce sperm or egg. So in the case of males, these would be the testes, which would create sperm. In the case of females, this would be the ovaries, which create eggs. So because of the fact that your sex cells will go on to create offspring, right? Sperm and egg will leave you to, well, maybe, to create offspring. Those are heritable. So it's any mutations that occur in the sperm and the egg or in the cells that will give rise to the sperm and the egg are heritable because they are passed directly to the offspring. Any mutations that occur in somatic cells, so the cells that make up your skin or your muscles, etc., those cells are not passed to your offspring. And so for that reason, genetic mutations that occur in somatic cells are not heritable. So that's when we get to this slide, which kind of also discusses the overall impact of these mutations. So starting on the left, where we're looking at germline mutations, let's say that you have a mutation that occurs in, this is like the testes, and so you have the sperm created by the testes carrying that mutation. So this means that the information carried by those sperm will contribute to half of the DNA carried by that organism. So well, in this case, because the sperm is contributing to half of the DNA, the sperm and the egg combine to create a diploid zygote, which then differentiates into a fetus. In that case, the zygote gives rise to all of the cells in the body, meaning that the mutation that was carried by the sperm will be present in every single cell in the adult organism. So that could be pretty drastic. Obviously, if it's survivable, it's not going to be that big of a deal. Why it says that half of the gametes carry that mutation is because remember that this process going from zygote to fully grown organism requires mitosis. We're going diploid to diploid. So all the resulting cells are going to carry that mutation. But the process of going from organism to their production of sperm involves the process of meiosis, where the genetic information in that individual is reduced by half. So that means half of the gametes would carry that mutation. Not all. Half of it are coming from the father or the grandfather. Half of it are coming from the mother, right? So in this case, right, if you have a mutation in the germline, that means that this organism, all of the cells in this organism are going to carry that mutation. On the flip side, let's say that neither of the gametes carry a mutation, so they form a zygote. None of the DNA in that zygote has a mutation. But what happens is at some point during differentiation, whether that be in a fetus or whether that be in a full adult, throughout the process of differentiation, there's a mutation. Because this didn't occur in a sperm or an egg cell, this is only going to affect the cells in which that mutation took place. In this case, what this slide is showing is a mutation taking place pretty early in development. It's kind of showing mutation occur in the location of stem cells that will give rise to like the shoulder. And so that means that all of the cells present in the shoulder would carry that mutation. Let's say you have an organism formed. There's no mutation here. We get to a fully formed adult. There's no mutation. But this person goes and lays out in the sun, and then one of their cells in their skin gets a mutation that causes a melanoma. That is a mutation that gives rise to cancer. That melanoma is not heritable. It will not be passed to its offspring because it didn't occur in a sperm or an egg that's going to be passed to offspring. So this is why germline mutations, germline cells give rise to sperm and egg, which are passed to offspring and are going to lead to every cell in the differentiating offspring. That's why they are heritable, but also more widespread as far as their effect. Whereas somatic mutations are not heritable because they're not taking place in cells that are inherited by offspring, but also their effect on the organism completely depends on basically what cell they're taking place in. Is it a stem cell or is it like a terminally differentiated cell, like a skin cell? And also at what point during differentiation it's occurring. Is it occurring early in development, like in this example where it's occurring in all the cells that give rise to the shoulder, or is it occurring late in development, right? Is it occurring in one skin cell and it leads to a melanoma, which is a very patch of cancer, specifically on the skin. Again, germline mutations will affect the entire offspring. whereas somatic mutations are not heritable, and it completely depends on timing during development as well as location. All right, I hope that makes sense. All right, and then finally, a lot of you were asking just for a little more information, seemingly out of curiosity, for nucleotide excision repair, so how we repair DNA mutations. So just a reminder of when we talked about this, what happens is if you have some sort of mistake in your DNA, so this is showing a pretty common mistake. This is a mistake that is commonly caused by damage from UV light. So this could be a mistake that could lead to like skin cancer. It's a pretty common mistake when exposed to UV light, and that's called a thymine dimer. What can happen is obviously RNA polymerase is not going to be able to transcribe this. DNA polymerase isn't going to work very well, and also just in general, the structural integrity of the DNA is not great. So what kind of happens is you have these proteins come along and check out the DNA. They notice this mistake, and they cause other proteins to come in here and basically say, like, we're just going to replace an entire portion of DNA. And what they do is they cut it out, and then they use the complementary strand to reconstruct a new strand of DNA that doesn't have a thymine dimer in it. So great, okay. So a couple of you asked how DNA damage is detected. So as this picture is showing, you actually have a bunch of proteins that their entire purpose is just to basically like check out DNA and look and check on it, make sure it's okay. These are collectively called damage sensing proteins. An example of this would be the DNA damage binding protein, or DDP. And basically what these do is they just kind of like survey the DNA, and if they detect damage, they're going to recruit the nucleotide excision repair machinery. So what kind of stuff are they detecting? We talked about thymine dimers, mismatched base pairs, thymine dimers. Any sort of issue with the DNA can cause disruptions in the DNA helix properly forming. So they will detect helical disruptions. These are actually really more efficient and effective in transcriptionally active areas of DNA. And that's actually because what would happen is if there's a thymine dimer and you have RNA polymerase going along your DNA, it's not going to know what to do with a thymine dimer, right? It's going to get to that thymine dimer and just be like, and stop. And so what a lot of these damage sensing proteins can do is they kind of like follow along with RNA polymerase. And if they see RNA polymerase like stall, then that can help them detect DNA damage as well. They can also do this to DNA polymerases as well. It's just RNA polymerases are a little bit more frequent, right? Cells are not always replicating their DNA, but transcription is pretty much always happening. So, so that is a really good indicator of DNA damage. So some of you asked, what if damage is not detected? And just to kind of give you an idea of how important this machinery is, if DNA damage is not detected, obviously you're going to get a lot higher of a rate of mutation, because this serves to detect mutations and repair it. So obviously without it, you have a higher rate of mutations. Without repair of mutations, you're also going to obviously get a higher rate of aging, which aging is, we didn't really talk about this in class, but aging really corresponds with your ability to repair genetic damage. And aging in general correlates with your nucleotide excision repair machinery kind of not working as well over time. It's not the only reason for aging, but it is a contributing factor. And then obviously, because genetic mutation is so correlated with cancer, you have a higher rate of cancer. So actually a little bit kind of like an interesting fact. The majority of our cellular functions are detected or discovered based on disorders when they're basically not functioning correctly. And this is the true for nucleotide excision repair machinery. We didn't really know it was a thing until we did kind of some investigating on a disorder called xeroderma pigmentosum, or XP. So it turns out that XP is a disorder that results from the body's inability to repair DNA damage. It is specifically a defect in the NER, being able to detect damage from UV light. And in fact, a lot of the proteins that are involved in the DNA damage repair pathway, if you were to look up this pathway, you would see a lot of the proteins are like XPA or XPB or XPD. That XP comes from the fact that we know that these proteins are not functioning in XP or xeroderma pigmentosum. So they're named based on this disorder because they were discovered through kind of understanding this disorder. What happens with this disorder? Obviously, you can kind of think about what happens when you get severely sunburned. You get kind of darkening of the skin, a lot more freckles. If you get damage that leads to kind of tumor formation or kind of really bad skin lesions, really bad eye damage, because if you think about how it hurts when you go in the sun, you like close your eyes. Your eyes are actually very susceptible to UV damage from UV light. Wear your sunglasses. But a lot of these kind of really, really horrible things happen because of really high sensitivity to light, what's called photosensitivity. So those pictures to the right are clinical images of xeroderma pigmentosum, sadly. But you can kind of think about with that disorder what happens when you get a really, really severe sunburn and you can't repair it, right? You can't repair that damage. Some other disorders that are correlated with inability to repair DNA damage. Another one that was kind of critical in the discovery of these pathways is called Cockayne syndrome, or CS. It has some pretty interesting phenotypes. Photosensitivity is also kind of like categorically a part of this because this pathway is so important for repairing damage in response to UV light. Cockayne syndrome and another one, which I'll talk about in a minute, also have kind of reduced mental capacity. Microcephaly is associated with CS, and some other symptoms that are very similar to progeria, which I'm not going to go into, but it is another genetic disorder, not distinctly related to NER, but the phenotypes kind of overlap here. Some of the proteins involved in the repair pathway, if you look more specifically, are going to be called CSA and CSB, and these were named because they were discovered as important proteins by discovering the lack of these proteins in this syndrome. So both those disorders, XP and CS, as sad as they are, were crucial to our understanding of this pathway. Another disorder that is characterized by a dysfunctional repair mechanism is called trichothiodystrophy, or TTD. But obviously this is going to also have a lot of photosensitivity. Ichthyosis, which is kind of like a toughening of the skin, almost like psoriasis, sort of like lesions, as well as reduced mental capacity. So pretty severe disorders, pretty distinct phenotypes, and hopefully what this does is it highlights the importance of this machinery in sort of the normal functioning of the body, because this is what happens when you don't have it. And I always like pointing out that as a way to kind of emphasize the importance of why we need it. So, all right. So that is basically what I wanted to cover as for kind of the common topics that were in the discussion board. As always, reach out if you have more questions or if you need something more clarified, and I will do my best to answer.